welcome. It is with a certain amount of trepidation that I stand here to introduce today's speaker. I say that because today's speaker is himself, excuse me, quite renowned, but also because he was introduced so effectively by our Dean of Science on Tuesday that I feel I have large shoes to fill. So as you can see from the title, today's speaker is Mu-ming Poo, who will give the second of this week's talks. This one more focused on neuroscience and neuroplasticity. Professor Poo received his BS in physics from Tsinghua University in Taiwan, PhD in biophysics from Johns Hopkins, and then after postdoctoral research at Purdue, basically bounced all over the United States at 3 different University of California campuses, first at Irvine, then at San Diego and Berkeley, and in between managed to also spend time at Yale University and Columbia University. He now is best known, I think, in Asia as the founding director of the Institute for Neuroscience in Shanghai, as a member of the Chinese Academy of Science, and as a member of the National Academy of Science. But these are just the dry facts of a person's career. And what I struggled with was how to really capture, for his introduction, what it is that makes him special. So I thought what I should do is go to the literature in keeping with the scholarship that was on display on Tuesday. So I turned to the ancient Chinese poetry, in particular the Lao Wai school of poetry, the southern branch, the Guilo branch.
Lao Wai?
Lao Wai. And I've always been particularly fond of that branch of poetry. There's a special type of poem known as the lyrique, which Mu-ming knows very well. So I found one that I thought was particularly relevant to today, and I thought I would share it with you. So please listen and stop what you do. I have quite a treat here for you. A bright shining star who has come from afar to share his remarkable view. On Tuesday, to standing ovation, he spoke to us on innovation. A philosopher king is our friend Prof Mu-ming. His words were a true inspiration. But one talk for Mu-ming's not enough. So to distill it all down is too tough. So today is talk 2, where he'll give us his view on plasticity and other stuff. But now I've said more than my share. Any longer just wouldn't be fair. So without more ado, please greet Mu-ming Poo and let your applause fill the air.
Thank you, Carl. That was wonderful. I remember in the Yale days, we have this annual neuroscience meeting where what we look forward to is not only science, but Carl's limerick. This is one of the display of old tradition. Today's topic is scientific, but in a very general sense. I'm not going to give you the latest finding from this field, but to summarize what I feel is a very important aspect of the brain, that is the plasticity. Now, what is plasticity? Let's define very clearly in the beginning. It's the ability of the nervous system to adopt a new functional and structural state in response to extrinsic and intrinsic factors. Now, this is a complicated statement of the fact that brain can change in response to outside influence and inside influence. Now, this change of the brain tissue is very special to the brain because many other tissue also changed, but that change is trying to keep the homeostasis. when you respond to the environment, the conditions change, the tissue will try to maintain its original state rather than adapt a new state. Right? But this property of adapt a new state is a plasticity that's exactly opposite to homeostasis. Now, why does the brain want to do this? Well, this actually is the reason that brain has the capability of learning to adapt to the environment. Because the environment has changed, the plasticity allows the brain to adopt a new state by learning, by memory, so that they can respond to the environment better. So this is the plasticity. Now, there are many different types of plasticity. Normally, the plasticity we can think, the best example is learning and memory. Now, when you go through today's talk, you remember something that might keep in your memory for a long time, I hope, right, if I'm successful. And that could even keep for years if you really take my message to heart. Now, this ability is the normal plasticity, right? Because to keep the memory for a long time, you have to change. Your brain has to change somewhere, has to store this memory. And the long-term changes must be structural change that are stable. There's also impaired plasticity. Now, there are mental retardation, there are all kinds of disease where the brain cannot adapt to the environment. That's the loss of plasticity. We can call that a pathological state. But sometimes after injury, the brain will also change. It adopts a new state in order to help to accommodate the new after the damage. Now, this new adaptive change is helpful to the animal to survive. And that type of adaptive change is different from normal plasticity, as I'll describe further in a minute. Sometimes the adaptivity, the plasticity is bad, right? The best example would be addiction. Right? That's a new circular adaptive state. You just cannot get away from it. You always need to have this particular addictive substance or behavior. So that's a maladaptive plasticity. So there are different types of plasticity. Some of them are directly related to brain disease. And learning the plasticity in the laboratory is very important for our understanding how to cure brain disease. And so we recently wrote a paper to summarize different types of plasticity that one can translate what we understand in the bench to the bedside. So let me give an example of the plasticity in the brain. Now, in our brain, different region of the brain is devoted to different tasks. In motor cortex, different part of the motor cortex is devoted to controlling muscle in different region of the body. Now, this map is a very distorted map. The region that we require more, we need a higher resolution in our motor action. We devote more cortical area to control it. Right? So the proportion of the cortical tissue devoted to different motor function or sensory function is directly proportional to the importance of that part to your body. Right? So in human, in particular, our hands and our face are particularly important. It's the hands, the flexible and capable hands that create the civilization in a way. And the social interaction depends on the muscle on the face, expression on the face. So it's very important to control the face muscle. So this map, everybody has it, but it's different in a minor detail. For example, we know that this map can be plastic. For those violin players who use their left fingers a lot, their right cortex, motor cortex, the area control their fingers are enlarged. And if you start to practice early in your life, you have larger cortical area devoted to your left fingers than starting practice later in life. So there's a activity-dependent enlargement of the cortical map. And this is a very dramatic macroscopic plasticity of the brain. This can be very quickly examined in monkey, for example. If you train a monkey only to use the middle 3 fingers, the region devoted to these 3 fingers will be enlarged and take over the region adjacent finger. This can be demonstrated quickly within a few months. There's also remapping of the maps after injury. There's after amputation of your arm, for example, there's a sensation of the missing arm. It's called the phantom limb syndrome. You can still feel the presence of your arm, even if the arm is amputated. This is well known. But the reason is that the region in your brain that control the fingers, control your arm, that feel your arm is still there. Now somehow the sensation in other parts of the body trigger the firing of the region that originally controlled your arm, then you feel as if the arm is still there. Right? So here is an interesting remapping of these map like controlling the fingers. Rama Shantung, a clinician, a neurologist at UCSD, he found that some of the patients, on their face, there seems to have finger feelings. So you touch the face, different region of the face mapped oddly the finger 1 to 5. Right? So somehow the finger, the cortical area that control the fingers now feel there's input. Although the arm is gone, there's no fingers around. Right? So the way to explain that, this is the best way to explain it. The face area are very close to your finger area in the cortex. Normally, the sensation from the face goes to the face area, so you feel things on the face. But when the input to your finger, to your arm and your fingers is gone, these cortical cells are deprived of their input. They start to receive sprouting of axons from the adjacent area. So when you touch the face, you will not only feel it's a facial feeling, but you also you feel that there are input to your finger area. Your finger is being touched. That's why the finger are mapped onto your face. This is this is a very gross remapping of axon sprouting. The existing axon just cover new areas. So this is the clinical phenomenon. Now to further enhance your feeling about plasticity, Rama Shantung had another dramatic case. Some of these amputated patients feel extreme pain on their amputated arm, even though the arm is not there. Now some of them feel that their fist is always in tightly clamped position to give them very painful sensation for a long time. Could not release their hands, or even though the hands is not there. Right? So he very ingeniously thought about a new way of helping this patient. He take a box with a mirror in between. The right hand is still okay, so he asked the patient to stick his right hand in the box. The patient look at the mirror as if his left hand is still there. And then he asked the patient to release his arm, both hands together, practice releasing his hand. Right? So the patient would feel as if his left hand is still there. So he gradually, after practice for a few weeks, the pain of the missing arm is gone, the fist is released, so everything's fine. Right? So this is a very dramatic finding for the changing the brain simply by a visual input that regain the original sensation. That's a normal sensation. Now, why this has happened, now, there's no good explanation. One can imagine that this is a plasticity of the visual somatosensory feedback loop. When we normally touch something, we do some motor action, our visual feedback is very important. Right? Now, by regaining that visual feedback, you regain the normal state of your brain that has been adapted in a new state during the amputation. Now, that's the very rough way of saying it, but the fact is a simple input to a pathological condition can greatly change the structure of your brain. That can be a approach for therapy of many of the neurological problems. Right? So this is a very good example of plasticity. So now how does the activity change the brain? There's something must be changed. Now it's very clear from all we know now, the activity of the brain is carried by electrical activity. Right? Your sensory input, your motor action, or your cognitive function, all associated with electrical activity in your neural circuits in your brain. Right? This experience must have changed something to put the brain into a new state. Right? So the general consensus is that the way that this activity does modify the brain is to change the structure or the function of the synapse or the neurons, because that's the basic structure of the network. Now, most people think that plasticity is at the synapse because this is the region that much more susceptible to change. It's much complicated in structure than the neuron or axon, the conducting part of itself, because that is less flexible. So the most flexible and changeable place is probably at the synapse. So all this activity, you want to understand the plasticity, you have to understand how activity can modify the synaptic structure in particular. Now, once that structure is modified into a new state, what you gain is you have learning and memory, you have change in your cognitive function and in your behavior. Now, how does this activity change the synapse? 60 years ago, a Canadian psychologist by the name of Donald Hebb proposed a theory. He said that if we have a neuron that fires the other neurons, if these two neurons keeps firing together, that means one neuron is exciting other neuron in a correlated manner. Activity goes in this cell, immediately to activity in another cell. So their connection between these two correlated firing cells will be strengthened. While if the other cell were not fired, while this first neuron is firing, then this activity are not correlated, their connection may be weakened. Now this idea that correlated firing, firing at the same time, was a very important proposal, because we think many of our experiences are related to timing. Because I remember an event, I saw your picture, and your face giving me firing of cells in my brain simultaneously, so I recognize your face. Now this is simultaneity of firing of different neurons. The correlation is always an important part of the memory. So correlation as a basis for changing the synapse is a very useful idea. So people say that the simple way of putting this is that cells that fire together, they will be wired together. Their connection will be strengthened. Now this is a very useful rule, as I'll show you in a minute why they are very useful. But does the activity change the synapse? Activity is carried in the brain by this firing of pulses. Now the pulses of electrical current flow through the axon, long axon. The activity is carried by waves. This is very slow waves compared to electron flow in the cable. It's an ion that carries the current that goes in and out of the cell. And that positive ion flow into the cell creates a wave of activity. And that propagates along the axon, a very long axon, to the end. Now, once they reach the end, it does not go directly to the next cell. They actually release chemicals we call neurotransmitter. This transmitter opens channels in the other cell that allow the current to change, the potential to change across that membrane. That gives you the signal in the next cell. So activity goes from one cell to next. That's how the electrical current flow in the system. Now, the unit of this signal is what I call a wave of activity of ions across membrane. It's called action potential. You can recall that action potential, put an electrode into the cell. You can see that the inside of the cell is a negative, normally negative potential. When the cell is fired, is active, they fire a very short pulse of 1 millisecond pulse. And that's the unit of signals in the nervous system. And the signal is carried, they code information by both the frequency and the timing. Now, if you have a different size of stimulus or activity that causes the depolarization of the neuron, the nerve cell, the timing of the firing is important. That codes for the initiation of the signal. The frequency is proportional to the amplitude of the stimulus. So the coding is the frequency of the pulses as well as the timing of initiation of pulses. Now, the timing code and the frequency code are the two basic way of coding information in the brain. The synapse, this is the most important region of the brain. Those of you who are not biologists, knowing synapse, 突触, is the basic concept of today's talk. These structures are called chemical synapse because the signal from one cell to the next is mediated by release of chemicals at this junction of synapse, called synapse. The reason that the activity doesn't go straight to the next cells, which sounds easier to carry activity, transmit activity, but the chemical synapse is so complicated for a reason. First of all, the amount of transmitter we can release can change. The amount of receptors that receive, or ion channel that receive the transmitter can change. And the nature of the ion that flows through this membrane can be positive or negative. It can be a positive ion or negative ion. So there are a lot of variability and a lot of capacity of changing the transmission as well as changing the sign of the signal from positive to negative, right? So chemical synapse is particularly useful in storing information by having this complicated structure. So what we said, the activity can leave an imprint in the brain to change the brain to a new state. The plasticity is mostly involved in activity going through this synapse, changing the efficiency of this transmission. Right? The amount of the transmitter release or the number of receptors that control the postsynaptic membrane currents. Now, this first evidence for this activity that can change synapse actually happened here was found in China. In the 1941, in Beijing I should go back. In Beijing Union College, Beijing 协和医学院, Feng Pei, a very young neurophysiology, returning from UK after PhD training, to Beijing to join the physiology department in Union Medical College, which is the first graduate school established in China by Rockefeller Foundation. Now, Feng Pei was doing the really pioneer work on the synaptic transmission, using the neuromuscular junction from the motor neuron to the muscle cell. That neuromuscular junction is a model synapse for studying the function of synaptic transmission, especially in the early days. What he found in the 26th paper of his series, published in the Chinese Journal of Physiology, which is still existing today, actually in Taipei. This journal was founded in the early 20s by Licht, a chair of the physiology department where the Feng Pei joined. Now, in those early days, Feng Pei's paper is widely read across the world. Now, Chung and Huxley, as you know, who got Nobel Prize for for working out the ion channel, the ionic basis of the action potential. In his memoir, he said that time in 1941, they were all joined the Army or Navy for defense of the UK. All the young scientists are all in service. Hodgkin was doing doing research on radar, low altitude radar. He said the one thing that he expects every month is to wait for the C-mail of Chinese Journal of Physiology, where he can read Peng-Deibei's paper on neuromuscular junction. One of the highlights of his scientific activity during that war period. Okay, that's the Peng-Deibei's work. Now, this happened to be the first finding of plasticity, because Peng-Deibei applied high-frequency stimulation to neuromuscular junction and measured the end plate potential, the synaptic potential triggered by every action potential in a muscle cell. The amplitude of that end plate potential goes up after high-frequency stimulation. It goes up and then goes down, return to normal. And that increase is lasts for about 10 minutes. Short-term potentiation of transmission by high-frequency stimulation, which is a phenomenon that activity produce something in the synapse that lasts for 10 minutes, a short-term memory. So this is the first discovery of synaptic plasticity in 1941. This is actually before Hebb proposed his model of synaptic learning. Now, this is a neuromuscular junction, but 30 years later, another very important discovery by Bliss and Nomo in central nervous system, a similar phenomenon. More strikingly, that in this discovery, we're now called long-term potentiation, LTP. Right? Remember this is the key word of this lecture. LTP is a phenomenon where you give a high-frequency stimulation of synapse, then the efficiency of transmission will last for a long time, so-called long-term potentiation. The long-term potentiation is even more striking than Peng-Deibei's short-term potentiation. It's lasted for hours, even for weeks in some places. Now, this long-term potentiation is now considered to be the base for long-term memory, at a cellular basis for long-term memory. So the discovery of the LTP, long-term potentiation, and later by Masao Ito in Japan, the long-term depression, the opposite phenomenon, that you fire not with high-frequency stimulation, but low-frequency stimulation, the efficiency drops and persistently maintained at a low level. It's called long-term depression. So both potentiation or depression can be induced by electrical stimulation of a different pattern. Now, this is actually fits well with Hebb's idea, because high-frequency stimulation, the postsynaptic potential can accumulate because it's high frequency. You can sum up and potentiate, cause accumulation of depolarization, then fire the postsynaptic cell. So high frequency stimulation trigger pre and post correlated firing. That's what cell fire together, wire together. At low frequency stimulation, presynaptic cell is firing, but postsynaptic cell are not able to accumulate because it's low frequency, cannot accumulate depolarization. So they decay without going through the threshold to fire the cell. So you have a presynaptic cell firing, postsynaptic cell not firing, uncorrelated activity. You get depression. So correlated versus uncorrelated is a good way of looking at this fit with Hebb's hypothesis. So this is the functional change of the synapse. However, where is the synapse? Well, the synapse is between the axon terminal and the postsynaptic receiving structure called dendrite. Now, interestingly, this for most excitatory synapse in the brain, this synapse is actually occurring in a very interesting structure. The postsynaptic receiving structure, dendrite, actually have a protrusion called spine. Dendritic spine is where the nerve terminal will make the ending. Now, why? Why this so complicated structure? Why can the input directly ends on the postsynaptic dendrite and then fire the cell body? That's much easier, right? So this complicated structure was discovered in the 40s by when people look at even earlier. Actually, I think Cahal also in his drawing also showed these spines. But this early discovery of spine, nobody knows why they are there. Now, the person who proposed this structure has a meaning. Is a person you should all know, Zhang Shantung. Now, some of you may know. Zhang Shantung is the grandfather of modern Chinese brain science, the head of the Shanghai Brain Science Institute, the precursor of my institute, Institute of Neuroscience. We actually stay in the same building. And in fact, in his late stage, he is still occupying an office next to mine until he was 100 years old. Zhang Shantung proposed in 1952, before he came back to China, in a Coast Harbor Symposium meeting, he looked at this structure of spine. He said they probably serve a function of controlling the input to the dendrite. Now, in this paper, he said this structure actually, because they produce a high ohmic resistance of current flow, during the neck is very difficult for current to pass through. So by changing the structure or the neck thickness, you control how much current flows through to the dendrite to be summed up by the postsynaptic cell. So but the structure of the spine become a way of controlling the weight of the synapse. This idea he proposed, nobody paid attention to it in this symposium review until, again, 30 years later, the person who dig that out is Francis Crick. Now, you know Francis Crick, the person who discovered the double helix. In the early 70s, late 60s, he said, well, we understand how genetic material works. So the more important thing is to now understand how the brain works. So he started to become a neurobiologist. And the way he does it is not doing experiment on the brain, but sitting in his office, just think about how the brain works. That's how theoretical biologists, very few of them, like Francis Crick can do. So he was thinking how the memory can be stored. He looked at the spine. He also feel it's strange. It's clear that spine, he said, if the spine can twitch like a muscle, can change its shape, it might be useful as a way of storing memory. Now, he wrote in a review. And in this review, the first paragraph of that review, he said the most interesting suggestion about what spine work is by T.H. Chen in 1952, that the electrical resistance of the neck of the spine reduced the weight of the synapse. That's Zhang Xiangtong's suggestion. And based on this, he suggests that spine morphology is a way of controlling synaptic weight, in addition to the function. LTP and LTD is the functional transmission can be changed. Now, in addition, if you change the structure, you have another way of changing the transmission efficiency. Right? So now let's look at what we now know about the neural basis of learning and memory. Now, the first question people would ask, where's the memory? Now, we know they are probably at a spine, a synapse somewhere, but where in the brain? We don't know. This question is so-called, where's the engram? Engram is the memory trace. It's a question that I asked for now 100 years by the neuroscientist. The question is, where are these engrams? In which brain region do you store the memory? Right? For example, today's talk, where do you store the memory? We don't actually know. Which neurons and which synapse store the memory? Because there are millions and hundreds of millions of synapses for every activity going through your brain. So many synapses are involved. Where are they? That change it into LTP or LTD or change the structure. Now, this is the question. It's still unknown. We know the memory is stored, probably in the synaptic strength, LTP and LTD, right? It's possible there's also change in structure so that the connectivity is changed. The question is, but how much weight is the memory stored in the function versus the structure? That we don't actually know. In the adult brain, when we have a memory, is the memory change the existing synaptic connections or are actually making new synapses? That we don't know. We don't know the relative weight of these two mechanisms. We don't know how the memory decay with age. Now, in old age, is the memory decay? We know memory will decay, but is the decay the ability to form a memory or the ability to consolidate the memory? You can take a memory, but it only shorten, you would forget it. Right? These are the questions that are still unknown. So actually, we know very little about learning of memory, except we know the building blocks of memory is probably those tiny synapses that changes the function or structure. So I'm going to show you a set of experiments recently from my lab on looking at a structural change in the adult brain. What happened? Now you can look at the synapse and this dendritic spine and their connections by so-called two-photon imaging. In this image, you open a window on the head of the mice. You look at the fluorescent labeled cells. You can look at the structure in living state, the axons and dendrites and their connections. You can actually go back to the same place within days and weeks to find exactly the same place, same dendrite and the same spines, and see whether they change. And so now the question is, if you now produce memory in this region, that, for example, involved in particular memory, you ask what happened to their connectivity. Right? So, does the memory, long-term memory, involve forming of new synapse, structural change? So we use this auditory field conditioning. It's a Pavlovian type of association learning. You give a tone, a certain frequency, rather complicated tone to give to the animal. Right at the end of the tone, you electrically shock the animal. You pair these two stimulus for a while. Then if you only give the tone, the animal would freeze, as if there would be shock would be coming, so-called field memory. Now, the control for this is that if you don't pair these two stimulus, they don't learn. The sound will not produce any freezing behavior. Right? Very clear difference that the pairing of this shock with the tone gives you fear response with the tone alone afterwards. So this behavior, very robust behavior, involves auditory cortex. So we go through the auditory cortex and see whether there are structural change. Now I want to go quickly to, you can label the input to the auditory cortex from a subcortical structure called amygdala, which is involved in the field conditioning. Their output, their axon is on the cortex. So here's the region of cortex that we look for their structural change that potentially code for the memory. So it turns out that if you follow the axons from this region to the cortex, the axon, the place they form synapse is on this axonal swellings called bouton. This axonal bouton actually can appear, form, or disappear in the adult brain, but at a very low frequency. Normally, it's about, in the control animal, it's about less than 10% of the new synapse would appear in 2 hours. After 3 days, maybe 10% of new synapse would form. A very small percentage of formation of these new boutons. But if you have animal conditioned, there are significantly more new bouton forms at the auditory cortex. The bouton that disappear doesn't seem to change. There's still some disappearance going on that's similar to control. So the net effect is that learned animals have more net number of synapse, so no new additional synapse at this region. The other, if you label the input from other region called the prefrontal cortex, you don't see this phenomenon. So it's a very specific subcortical input to the cortex that change. You can also look at a postsynaptic cell. That's the spine. Remember the spine where the presynaptic axon comes in and makes contact to the spine. Now, the presynaptic axons are not labeled. If you only label the postsynaptic dendrites, you can see these spines. And the spine also increases over the first 3 days after conditioning. The spine also disappear. Some spine disappear. And the disappearing of the spine doesn't change. So this is a layer 5, the dendrite from layer 5 cortical neurons that have these postsynaptic protrusions that appear to be more after learning. Now, the Neoto3 neuron doesn't have this phenomenon. Again, it's a specific input that causes postsynaptic changes. Now, we have presynaptic change, we have postsynaptic change. It's better that we have labeled both at the same time so we can actually identify the axons, which is now labeled green, and dendrite that labeled red, from defined regions, right? Axon from defined region with a defined postsynaptic cell. So we actually know what synapse they are. Now, if you do that, it's a very difficult procedure, but finally we accomplish to do it. We can actually monitor these pairs of synaptic pairs and look at their changes. Some like this one is stable over 3 days. Most synapses are stable, but some synapses will grow. Now, for example, here there's the axon, the green boutons form on a spine protrusion within 3 days. Now here's one that disappeared. The spine disappeared, the red spine disappeared. Here's this new spine, a new bouton formed. So there are new formation and elimination of the synapse. Now this is low frequency. You are seeing the changes which are in a kind of normal control. It's 10% changes overall. 10% new formation, about 10% eliminated. So the steady state turnover is this low percentage. When you have a memory, what you have is in this particular case, you have increased formation of new spines and new boutons, the new synapse. The old synapse elimination remain the same rate. Right? So the net result is that the long-term memory associated with the field conditioning produces new synapse. Right? Even though it's not that many, it's from 10% to 20% increase. Now, the other interesting thing is that if you look at this change, you have here's a case where the presynaptic axon going through the in touch with the postsynaptic cell. Within 3 days, a new spines form that make the synapse with presynaptic axon, bouton. Now this bouton is probably have a synapse, has a postsynaptic partner, a spine which are not labeled, because we only do sparse labeling. So what you have is an old synapse now have a new element of spines add on top of it. You will have another situation where you have a spine that normally have its own partner, but with time, a new bouton is formed on that old spine. So we add on top of the original synapse. Now this cell, sometimes you can see the original pairs and then see the new pairs forming on top, new spine on top of the old pair of pre and postsynaptic structure. This is, again, a new bouton on an old pairs. So what you are seeing is that all the new formation, it turns out 50% are new spine on the old bouton. Another 50% or so is new boutons on old spine. Rarely, less than 1%, you have a completely new connection form. That is new bouton and a new spine. So what you have is an adult formation of new synapse that are adding new elements to the pre-existing synapse to increase the efficiency of connection between the presynaptic cell and postsynaptic cell. Now that's interesting because this is actually what's happening in the adult brain, that it's a very energy-saving way of increasing efficiency. It's not making de novo new connection. It just sprout a little bit to add a new partner to your original synapse. So you have a multi-synapse formation on the existing synapse as a normal way of adding new connections in the brain. Now, even though that happens only still not a very widespread, it's only a small percentage of spine that actually change. So auditory condition field memory is stored in part in the auditory cortex, and the memory storage involves structural rewiring of the synaptic connection, in addition to LTP and LTD, functional change. And these changes, rewiring, involves adding new synaptic partner to existing synaptic elements. A very efficient, energy-saving, space-saving. Right? There's very crowded space in the adult brain. You want to add new synapses, it's not that easy. This is the way that new synapses are made. Now, this is not what happened in early development. When the baby was born in human, you have very little arborization of your dendrites. Now, newborn baby have cells all born. All the neurons are born. What happens in postnatal first 2 years is a tremendous increase in the processes, the dendritic processes and axonal processes that establish a very complicated network. That's during the early, the postnatal period. Now, this excessive growth is unlike in adult brain, right? In adult brain, you have a very small percentage actually are forming, new connections are forming. Now, why is this, right? There's a purpose for this. If you look at the spine, which is a representation of the number of synapses that's formed in the brain, during the early period after birth, there's a tremendous increase of new formation of connectivity, connections, establishing the networks. Then there's a reduction of number of spines, right? There's a pruning of the spine. Now, why is that? there are a lot of connections made in early period. Then you eliminate some of the connection. Gross elimination, large number, now 30–40% of synapses are gone. Because you are learning, you want to consolidate the use for correct synapse. The early formation is a random network that has not been tested. So you want to learn, you want to crawl, you want to stand up, all the motor circuits has to learn. And then you establish a useful, correct network for your function. So this pruning process, very excessive pruning during early development, is a way of consolidating the correct connections. Now that's an important developmental plasticity, right? These are totally different from the adult plasticity, which involve very small changes of the synapse. Now, you have a normal pruning. If you have a schizophrenia, this apparently there's more excessive pruning of the spine. Autism seems to have a insufficient pruning of the spine, suggesting the network is affected during development. Right? So this is plasticity that's abnormal plasticity that leads to pathological condition. Certainly, there are spine degeneration. In Alzheimer's patient, there's a certain drop. It's not a gradual drop in a normal case, but a certain drop in at a disease onset of the Alzheimer's. That means the synapses are degenerating. So your memory function is drastically reduced. So we talk about the HAPS postulate, HAP learning rule. The correlated firing, the cells that wire together, fire together. This is very useful. Now we know this fire together, wire together, including not only a change in the efficiency of connection between two cells, but maybe in addition, new connections are made. Right? That's a structure, both structure and functional connection. Now, why is this interesting? When HAP proposed this idea, he was trying to understand how we keep our memory. For example, how do we keep the memory of a circle in our visual field? He was imagining that this circle in your visual field, if you want to remember it, this circle must excite your visual neurons, a group of them, simultaneously. Now, for example, these segments excite this neuron, that segment excite this neuron. When the circle is present, when you are exposed to this visual experience, you have a simultaneous correlated firing of a specific set of neurons. He calls cell assembly. These cell assembly are excited at the same time. The connection is strengthened. So this strengthening of connection among a defined group of cells is where you keep the memory. Now, what do you represent? How do you represent the memory? Well, if this set of cells is fired again simultaneously, that's a reappearance. That's a recall of your memory. And this is how he imagined the brain keeps memory and how you record the memory. Right? This is HAPS theory. It's called cell assembly theory. Now, for example, if you see a partial visual cue now, you only excite a few cells. But since they are now highly connected, this partial stimulus would excite the entire set of cells. The result is you can recall, by exciting that assembly, you recall the memory of the circle. Right? This is how HAPS imagined the assembly. This is a very useful idea because this set of cells can code a feature, a visual feature. In this particular case, this is a circle. Now, there are other visual features. There are colors. There are if you have an object going through your visual field, you want to code, you have an impression and a visual perception of color, you have a texture, the contrast, contour, all kinds of features. Each feature is coded by a group of cells, assembly. So the entire object is an assembly. You will repeatedly see in this object. You will strengthen assembly of assembly, groups of a group of cells that link to another group of cells, all linked together to form a huge super assembly that codes for this object. Right? So your memory of an object is a cell assembly, a nested network. All the memory is kept at the connectivity between this group of cells. Right? This is the HAPS way of understanding an object. Well, how do you remember your grandmother's face? Well, your grandmother's hair, nose, eye, all have its features. Each one kept stored in this assembly that codes for particular features. And then the entire assembly, because of the presence of your grandmother for a long period, you kept this assembly, which are interconnected with strengthened connection. So when you see a partial feature of your grandmother, you can actually evoke the entire grandmother's face in your brain. You recall your grandmother. Now that actually can be demonstrated in monkeys. Monkey can recognize face very well. Human face, monkey face. Now, if you go to the region that codes for the face area of the monkey, in the temporal lobe of the monkey, some cells actually respond very well to the monkey's face. Now, you have a partial feature. Sufficient partial feature will also evoke this cell. But if the feature is not enough, you don't evoke that cell. You have, in these two partial feature of the face, insufficient evoked cell. So in HAPS idea is that all these partial features, each one's activate a subset of your face. If sufficient subsets are activated, the entire features will appear in the monkey's mind. And this cell that's been recorded happen to be excited only when the entire, a sufficient number of features are excited. So they were part of this assembly that been recorded. And in fact, there are thousands, probably even hundreds of thousands of cells that the entire assembly of them actually codes for all the features of their face. Right? So this is how I understand the storage of memory. So can you tell what you see on the graph? A dog. Some people see a dog. Other people, many others didn't see it. Okay, those who can see a dog here is the head of a dog. It's a dog with a white dog with black spots on the body, right? So this is a neck. So this is a dog face. This is a That's a dog. So why can you recognize it's a dog? It's only a very partial feature that's in your brain. The reason you can recognize is those who can recognize this actually have seen this type of dog, which is Dalmatian, right? The dog with a black spot and a white fur. This type of dog, the image of the dog was in your brain because you have seen before. Now you recall this image only with partial stimulus. That if I've never seen a dog, you will never recognize this because you will never have established assembly in your brain that can be recalled. Right? The firing cannot be recalled. So now memory is much more complicated. There are a lot of events you will remember, not just as simultaneously presented to you. Music is not like that, right? So you remember a tune. So I'm going to play a game with you. Now name that tune, the first tune. A famous composer wrote that. Can you remember that? I would doubt you can. So the second tune. Most of you will recognize that, right? The rudimentary piano lesson for kids who study piano would play this. Beethoven for Elise. 给爱丽丝. This turns out that these two pieces of tune was the same score, but the first tune is the computer plays backwards. Same interval, same score. You don't remember. You cannot recognize it. Okay, that tells you that memory is temporally specific, and it is a sequence of events, and the interval of events are critical in your memory. So you have this long-term memory of this music. So how do you understand that? It helps partially saying that if you have simultaneous visual image, simultaneous excitation of the cells, you set up an assembly that can be evoked. Now, how do I evoke a sequence of events in your memory? So now let's take the LTP that LTD was saying. That the basis was the synaptic change. Now, in the 16 years ago, a number of laboratories, including mine, discovered a phenomenon called that of LTP and LTD that depends on the timing of pre- and postsynaptic firing. So here is the input to the cell. Postsynaptic cell have a depolarization, then immediately followed by spiking. Now, if this input is followed by spiking of postsynaptic cell, it gets LTP. It becomes potentiated. If you have an input that comes in right after postsynaptic cell has fired, this input will be depressed, LTD, entire opposite. Now, if you look at the HAPS original correlated cell that fire together and wire together, this pre- and postsynaptic firing, this is presynaptic firing, this is postsynaptic firing, they were all 10 milliseconds apart. They were all similarly correlated. But the firing sequence, presynaptic before postsynaptic, gives you LTP. The reverse sequence gives you LTD. So this is called temporal specific HAPS learning rule. It started to become very popular after late 90s when this was discovered. And this says that if you pre-firing is before post, you get strengthening. You reverse the order, same interval, you get weakening. Now this is very interesting because our interval is the same, just like the demonstration I've shown you. You reverse the interval and reverse the sequence. You have an opposite effect, right? So this is now called spike timing-dependent plasticity. So we see how this works. Now see, we have the simultaneous excitation of a group of cells, we call cell assembly. That connection will be storing a simultaneous image, right, without timing information. Now if we now extend that group of cells into a linear sequence. Now, every cell is connected to every other cell, but we are not exciting this group of cells together. We excite them in a sequence, in this along one sequence. Right? This would give you a firing pre-post. The synapse going downstream experience pre-post excitation. So this downstream going synapse are all strengthened. The reverse synapse are all weakened because they are this synapse, the post fire before pre-input, right? So you get a weakening. So a repetitive unidirectional sequential excitation will store the information in asymmetrically, not in all connection among all the cells, but in orderly potentiation within this assembly. Right? So once you have this established, you can recall this sequence, like firing a few notes in your sequence of memory of your music. You just excite only this cell in the beginning. They can trigger, because of enhanced connectivity, they can trigger the entire sequence. So if you remember well the music theme, I just play a few notes in the beginning, you can record everything afterwards, right? Because you have strengthened that downstream connections. You can fire cells in a sequence. Now, can you demonstrate this actually happening in the brain? So we have made a recording in the mouse brain with multi-electrodes and sweeping a ball across the visual field in only in one direction, from a start point to the end point. The cell, the electrode recording cell in the visual cortex, firing in a sequence. And then we train this repetitively for a while. And then we ask, if we now only fire starting points, can they trigger sequential firing along that direction? You store the sequential memory within a group of cells in the visual cortex. So I should show that this is a training. You see the cells fire, the cell responsible to this region in the visual field are firing in a sequence, right? So this is training. After a while, then you do testing. You excite in beginning or end. You see which one is likely to trigger a sequential fire in the trend direction, right? In the direction you have conditioned. You find that indeed you can actually produce more sequential firing when you, after training, when you stimulate the beginning of the firing. So here is the actual recording. This is 16 electrode recording, sort of random firing. Before, if you give a starting point a flash, you don't trigger a very clear sequential firing. But if you now give a unidirectional moving light spot across, you can see very good sequential firing recorded by 16 electrode, right? So you fire that first because that's what exposed to the excitation. Then there's a sequence. Now, once you have this training for a certain period, then you come back. You do the same flash as a control, like this. What you find is that the flash now, in the beginning, at the starting point, triggers a sequential firing. So you have a short-term memory of that sequential firing that's been trained. Right? So this is a short-term memory. It lasts for a few minutes, but this is saying that by the group of cells that have been sequentially fired can store information that's sequential, that can be recalled. So I'm going to end with a short final experiments. Let's talk about the cognition, because I talked a lot about synaptic mechanism. So what about cognition? What is the cognition, right? We talk about cognitive process in the brain. I think there are 3 levels of cognition we know. We recognize the outside world. This cognition of the outside world is one group of cognition that includes a sensory perception that we have talked about. Multisensory perception, you have a hearing, you have vision, smelling, all things usually has to be put together. You have to be integrated. you have attention, you have memory, you have categorization of outside objects. You have to make decision on what you've seen in the outside world. Now, all these are cognitions of the outside world. This is what most people in the neuroscience are studying, right? When they talk about cognition. But in fact, this is only part of cognition. There's another group, more important group of cognition. This cognition can be studied in many animal models, right? So from mouse to human to even worm and flies, you can study this cognition. But there's a cognition about self and non-self. Now, we all know, we and others, that cognition, self-awareness and empathy, theory of mind. I know what you're thinking. Theory of your mind. That's a theory. What the theory of mind means. I can have a theory of your mind. These are probably can study only in non-human primates because this is a higher cognition that lower animals doesn't have it. Finally, the third level of cognition that's the ultimate goal of the neuroscience to understand is the cognition of human language. No other animal can have it, no matter how you train them, right? Including our close relatives, the apes, the chimpanzees. Now, vocal communication with syntax and grammar, open-ended construction of sentences. These are ability, very mysterious and very important for human. If we want to understand the brain, if we don't understand how the cognition of language comes about, we are not there. We have not reached our goal. And this is, I think, the goal for the next generation, the young people around here. Now, this will be probably mostly resolved by the time you are researchers, now in 10, 15 years from now. But these two higher level cognition, we have barely begin. It's really rudimentary understanding. So I'm going to show you a set of experiments which you'll find fun. It's about self-awareness in monkey. Now, it's known for a long time that human and a few species of apes, chimps, they can recognize themselves in the mirror. Now, how do you tell? Now, if you have something on the face of the chimps or a little baby, they would scratch it. They look at the mirror, they know that's on their own face. Mirror self-recognition is a sign of self-awareness, the uniform way of reflecting self-awareness. Now, human baby usually do that between 1.5 and 2 years old. Now, if you have a baby, watch them. They cannot recognize themselves before, mostly before 1.5 years old. After 2 years, on average, they start to recognize themselves. Why? Is there a genetic program that the circuit certainly mature by 2 years old? Or are they trained? Are the parents taught them that they can recognize? Psychiatric patients and autistic patients some of them lost their self-awareness. So why? Now, what circuits went wrong? So it would be very nice to have a model where they don't have a self-awareness, then we can train them so that they can gain self-awareness so we can see what's happening in the brain. Right? So what are the species? Well, monkey. Monkey cannot recognize themselves in the mirror. So when you give a mirror, the monkey look at it, they will be frightened. No matter how long you train a monkey, put a mirror in their cage from the beginning, after they're born, for years, they will never recognize themselves. When you put something on the face, they look at the mirror, they would not scratch it. Some apes would do it, but not monkey. So the question is, can we train monkey to have this? So we put monkey in front of the mirrors. Here's sitting here, and force them to look at the mirror. And then we put a laser, a high-power laser. I don't know, high power in the sense that it gives it a little bit scratch feeling, itching feeling, so that they every time they see a spot in the mirror, they feel something is on their face. Right? They will scratch. Right? So this is we call visual somatosensory association training. So the point is to have them aware that something in the mirror on the face is on their own face. Right? This association, after a few weeks, this is done by Gongren and his student. This is during training. Now you have to feed them a little bit. In the beginning, they encourage them to do this. Okay, train 1 to 3 weeks, all monkeys, 7 of them. After this training, then you test them. They can do you test with a low power laser now. No feeling, no itching feeling. No food they will touch. Okay, so they would now recognize this association. So now I will put it back into the cage. So now this is a really critical test. This is how the standard gold test in the field, in the primate biology, is that you secretly put some dye on the monkey while they're not aware. Right? No smell, no odorless dye. And then you'll see what a monkey would do. So this is a trained monkey. They look at the mirror, and there's a stereotyped behavior. Looking, touching, and smelling. This 7 monkeys, five of them passed this test. There are 2 monkeys that are too stupid to go through this. When they put it in the cage, they lost interest in the mirror. It's not clear why they don't do it. Okay, there are only 5 out of 7 passed the test. Now, are they really recognize themselves? If there's another monkey in the cage that has something on their face, they look at a monkey face, will they touch themselves? Well, the answer is that this is a trained monkey. They see something on the other monkey's face. They would actually touch that monkey rather than touch themselves. That's not moving. Anyway, so another test. Now, if the 2 monkeys if 2 monkeys are separated by glass, both monkeys has marks on their face on the same position. Now, if they're just responding to some monkey face on the other side, they should scratch. And so we switch a glass wall versus a mirror, back and forth, defined time, and see what's the behavior. When they see a monkey face through the glass, they just want to play with each other. While mirrors in, they touch themselves. Right? So this is a monkey glass wall mirror experiment. Now, finally, this is totally spontaneous, no marks on the surface, on the face. See what they do. This is a trained monkey. Now, they are become very interested in themselves. Now, there are places that I've never seen before. Now they can use the mirror with spontaneous activity. Now, this is untrained one. Untrained monkey. They are sitting together, co-caged for 2 years, and it hasn't learned yet. The one that learn become better and better at it. Okay, so what's the implication? Rhesus monkey can learn mirror self-recognition. Human baby probably acquired the ability through learning during the first 2 years. Maybe the parents telling them, look, baby in the mirror, baby in the mirror. Now after a while, they start to learn. Now we don't know. You might want to do an experiment on your baby. See if you speed up their mirror self-recognition. Impairment of the self-awareness can be cured. Maybe you have a way of just like the mirror for the amputee, that you activate certain association, you change the brain in a way. Now, so monkey may have that circuit there. You just have to be trained, and now the experiment, you can see what's next, right? You want to go to the functional MRI to look at the structural imaging of the brain and see before and after training what happened in the brain. Okay, this is the end of the talk. I'm sorry I dragged long a while. Thank you.
Okay, I always wonder about why nature used the chemical synapse rather than the electrical synapse. Is that because the possibility of, how to modify the plasticity of the synapse?
Right. That's
now, if that is the case, can we predict in a different planet, they will have a different intelligent living system? Will you predict all the intelligent being, all the Martian and so on, they will all use a similar chemical synapse as we do, and none of them use this electrical synapse?
No, absolutely no, absolutely not. I mean, no, I don't know, but I can. But I would say that all these chemical synapse, all the our DNA code, all this will happen to us are evolutionary accident. Now we have a code of a triplet code now that's similar to bacteria. It's because now we inherited it. It's evolutionary inherited. Now evolution goes in a way by adapting. Now whatever accidentally mutated structure, we'll be seeing whether they fit with the environment. The chemical synapse happen to be chosen during the evolution to be an effective way. Now maybe other things will evolve that's even more effective. Now chemical synapse involve more than rather than the electrical synapse gets eliminated in the higher vertebrate because of plasticity. You offer the possibility of storing information. You offer the possibility of changing the sign of the signal. You can excitation comes in secreting an inhibitory factor that causes inhibition of the postsynaptic cell. So electrical synapse wouldn't be able to do that. A current from flow directly to the postsynaptic cell will have the same sign. So you lost the ability to change the sign of your signal. And all these are advantage to the evolution for the animal to adapt to the environment. So that's why it's selected. That's my opinion. Carl, you have a question?
I have a short question and a long question. So the short one is easier because when you talk about the new synapse, you never talk about is there any other protein being involved making a new synapse. Particularly in your model, it seems to be the activity will do most of the job. This is my short questions. My long question is, if I recall, when I look at a lot of old Chinese philosophy, we always mention that when the people last minute you pass away, you trigger the memory of many things in your brain. So could that, first of all, have anyone look at that, the functional MRI before we pass away for the last minute? So could that also, if they do, could that be a trigger on one single neurons to turn on all our memory back for a very short period of time?
Right. Well, that's very difficult to answer. The first question is easier. We don't know whether there's redistribution of original existing substance. We think that there are 3 days before we see, after training, 3 days for the structural change to happen. During these 3 days, there could be new protein synthesis that occur that allow the formation of additional synapse. It could be redistribution of resources without the original receptor gets redistributed and allow it to form a new synapse. When you have new synapse form on one side, does it accompany an elimination of resources or another synapse nearby in order to get that material? These are the experiments we would like to address. Now, so we don't have an answer to that. In terms of the refresh or the reappearance of memory, certain appearance of memory before death. I don't know how do you explain that phenomenon, but if memory recall involves what I said, the reactivation of cell assembly that represent different memories, then there could be activity happen in a person's brain right before death that trigger the reactivation of these assemblies. Now, where are the source of that activity? I don't know. Right? This is an interesting problem. There's actually an interesting thing that for brain science to work on is this consciousness. Right? There are a lot of people who had trauma who are in a vegetative state. Some of them recover, some don't. Right? What happened? Now, it's the vegetative state where you have no awareness, different from the sleep state. How do you monitor them and look at a difference? And that could be very important for the clinical treatment. So there's an unrelated problem, but it's a similar mysterious thing we need to know. Anyone? Shall please. Yeah.
Okay, so you were talking about a few cells associated with a particular memory.
Yeah.
I just want to know for a hard drive, for example, the information and the physical pixel, it's a one-to-one. Then once you use up everything, your hard drive is full, and then you cannot do anything. So in these neuron things, is it, you say, well, one particular involve multiple neurons. I just wonder whether that the same neuron also involve the other part of the information so that
Sure.
you don't have
Yes, same neuron can be involved in different, as a subset of different assembly. So you have assembly could involve, this assembly will involve overlapping neurons, right? And the neuron, each neuron doesn't have an identity. You don't know, when they fire, that doesn't mean that a single cell fire represents anything. It's a group of cells that fires, and the synapse they are activated together represent memory. So there's no one-to-one correspondence. I say these memories are this synapse, right? First of all, there are too many synapses involved in a particular memory to have that one-to-one correspondence. And secondly, all the information is not, there's no such thing as a memory cell. There's only set of synapses that codes memory.
So it's the connection.
It's the connection, yeah.
Okay, it's not
It's not not a single synapse, but a whole bunch of millions of synapses that codes one mind. That's the difficult thing of demonstrating where the synapses are. So people say, well, I want to activate particular synapse in the brain. Now, how do you do that? You cannot activate synapse because you don't know where they are. There are so many of them. You have to specifically activate them in order to activate a memory, right? So the recall of memory is best done in firing the cells that form the memory. Right? This is some of the experiment that the people you know, Tonigawa's experiment, are firing the cell that triggers the memory recall. But you cannot call those cells, which I have argument with Tonigawa. He calls the engram cell, but it's not engram cell. It's the connectivity that's actually stored in engram. That cell could be doing other engram when they're making different connections are activated. Yeah.
So if I suppose to have 100 synapse, that I have a different type of connection that corresponding to a different memory.
Yeah. Right.
Thank you.
Right.
Okay, maybe one last question. I'm actually not worried about Professor Poo getting tired, but I am worried about the audience getting tired.
Okay. Mu-ming, a very inspiring talk. Actually, you showed very interesting data. So that plasticity not just involve dendrites, as axon actually really they are plastic. Seems not many people study on axonal plasticity, right? So but I forgot the number you showed. So actually, really axon are as plastic as dendrites because you record a different event, right? They tend to send small spot. You basically in the adult they may not change structurally like dramatic,
Right.
And you also show this after trauma. For example, peripheral trauma or central trauma, things can dramatically change in the brain.
Right.
So is it possible, like say, in those extreme conditions, so the axon plasticity may play more important role than
So the extensive growth of axon in adult brain only occurs after injury. there's very little regrowth, and that's a big problem of axonal regeneration that's not happening because there are inhibitory substances produced in the brain. The capability of adult neuron to grow is very limited. However, when you have an injury, the healthy neuron sprouts. Healthy axon, when they see additional deprive the cells without input, they sprout out to support them, to make synapse. So extensive synaptic sprouting, axonal sprouting happen. Now, there's an axonal plasticity at a presynaptic terminal we call Bhutan. They form and disappear, form, disappear. There's a rapid redistribution of substance within the axons to form new synaptic connections, right? Maybe related to mitochondrial redistribution, stuff like that. So so there is axonal plasticity, but all happening at the presynaptic site, forming of presynaptic site. If you look at the transmission, you can say, wow, maybe you can change the speed of conduction by changing the number of sodium channels and potassium channel. So then the conducting efficiency is changed. Nobody has demonstrated anything about that, that you use plasticity in the ion channel density as a code for memory or for information coding. There's a disease where the conduction is disrupted, now demyelinated disease. Well, that's not the normal plasticity. Okay.
If there are no further questions, and maybe even if there are, let's thank our